Introduction to the electromagnetic brain. The purpose of this video is to transform the issue of consciousness in neuroscience into an electromagnetic problem. In today's world, it is widely accepted that the brain is an electromagnetic source. Now, in this series of videos, I intend to show where this idea comes and why there is a field called neuro-electromagnetics. Many of you may be familiar with the two devices, EEG and MEG. In other words, if we place sensors on the head, they record signals. The presence of these signals proves to us that a series of electromagnetic activities are taking place inside the brain. The connection between the brain and electromagnetism. This slide illustrates these sensors and the recorded signals. EEG sensors can measure the brain's electrical fields, while MEG sensors detect the presence of magnetic fields. As the results from these signals show, our brain has electrical and magnetic signals that vary over time and can be recorded from all areas of the brain. The primary goal of neuroelectromagnetics is to relate the temporal and spatial aspects of these signals to the neurophysiological realities of the brain, or the functioning of neurons. This understanding can have numerous applications. Brain disorders such as epilepsy, Alzheimer's, or Parkinson's can be diagnosed and monitored by creating functional images of the brain, and if surgery is necessary, the relevant information about the surgical site can be obtained. Human head anatomy. Now let's see where these signals originate from. To understand this, we first need to take a look at the human head and the position and structure of the brain within it. A general schematic of the human head is shown here. As you can see, the human brain is well protected inside the skull, which is a hard, bony structure. Additionally, layers of the scalp, composed of fat, muscle, skin, and hair, provide further protection for the skull. Beneath the skull, there is a dual-layer protective membrane called the dura, which encloses a fluid called cerebrospinal fluid, CSF. Within this space, there is also a double-layered arachnoid tissue and veins responsible for returning blood from the brain. The final layer is the pia mater, which covers the surface of the brain. Beyond these protective layers, we reach the brain tissue itself, which is broadly divided into two parts, the gray matter and the white matter. Let's focus on the brain tissue. This tissue includes parts such as the cerebral cortex, white matter, cerebellum, as well as the limbic system, midbrain, brainstem, and cerebellum. The cerebral cortex, which is the gray matter of the brain, is composed of two main hemispheres that are connected by axonal fibers called the corpus callosum. Additionally, the cerebral cortex can be categorized based on functional or structural classifications, and generally, it has topological divisions, such as its division into four lobes: occipital, temporal, parietal, and frontal, which are shown in the figure. Functionally, the occipital lobe is associated with regions in the cortex responsible for processing sensory information, including the primary visual cortex, related to visual information, the primary auditory cortex, related to auditory information, and the primary somatosensory cortex, related to bodily sensations. The region of the cortex involved in motor control is the primary motor cortex, and other areas of the cortex are associated with learning, perception, memory, planning, and movement. The cerebral cortex itself is composed of several different parts, each responsible for a specific function. Figure 5.8 shows these parts along with their functional roles. The figure shows the various regions of the cortex and the division of the brain into functional sections. In a lateral view, the two hemispheres within the brain are responsible for different functions. The left hemisphere is involved in the analysis of information and the control of sequential events, e.g., speaking, understanding the pace of others, reading, and writing. It handles part of the problem, while the right hemisphere is responsible for synthesis, combining separate elements to create a bigger picture, such as drawing a plan or reading a map. It is also involved in understanding the meanings of statements, and damage to this hemisphere can alter this ability. Although they are responsible for different tasks, these hemispheres exchange information through a bundle of connecting axons known as the corpus callosum. Behind the central sulcus are lobes responsible for learning, perceiving, and remembering. These include the occipital lobe, which processes visual information, the superior temporal lobe, which processes auditory information, and the parietal lobe, which processes movement and sensory information. However, these lobes also perform other functions, such as processes related to perception and understanding of the body. The frontal lobe is responsible for motor movements, such as planning strategies for action. The left frontal lobe, specifically Broca's area, is involved in controlling speech expression. The limbic system, a key area in the cerebral hemisphere, is crucial when it comes to learning, memory, and emotions. It consists of various regions, including the limbic cortex, hippocampus, and amygdala, the latter two of which can be found in the temporal lobe. The amygdala is related to emotions and behaviors like aggression, while the hippocampus plays a role in learning and memory. Individuals with hippocampal damage may struggle to learn new information but can still recall and remember past memories. Sensory information is received by the hypothalamus, including details about changes in the body's physiological state, such as body temperature. The hypothalamus also manages the pituitary gland, which is connected to the lower part of the hypothalamus, and controls the endocrine system, glands. Hormones function similarly to neurotransmitters, stimulating receptors to produce effects, but they work over much greater distances. When these hormones bind to receptors, their effect leads to physiological reactions in target cells, receptors in specific cells. The hypothalamus manages homeostatic processes through control of the pituitary gland and the autonomic nervous system. However, it can lead to more complex neural circuits in the cerebral cortex, resulting in learned behavior. The final figure also shows the various regions of the brainstem. The brainstem is composed of three parts: the medulla oblongata, which is responsible for managing heart rate, blood pressure, and breathing rate, the pons, which manages sleep and waking processes, and the midbrain, which controls body movement during fight-or-flight situations and when engaging in sexual behavior. If we focus on the tissue of the cerebral cortex, geometrically, it features grooves known as sulci and ridges called gyri. The gray matter, which is at its thickest between 2 to 3 mm, consists of 6 neuronal layers. Neurons, which are the primary elements involved in the brain's conscious activities, are located in these layers. Each neuron is composed of a nucleus, dendritic branches, and an axon that connects to other neurons at the synapse. Figure in right hand shows schematic view of distribution of the cortical neurons. The cross-sectional view of the simplified head model illustrates layer 3 and 5 pyramidal neurons, PN, and basket cells. The cortex can be represented by the crown, which is the top of the gyrus, the lip and the bank along the sulcus. Note that the axons of layer 5 PN in the lip and bank bend after they cross the boundary between the gray matter and white matter. In addition, it shows schematic view of the distribution of the PN. Note that neurons have relative orientations according to their locations. Neural electrical activity is driven by the generation and propagation of action potentials, essential for communication between neurons. An action potential occurs when a neuron's membrane potential rapidly changes due to the influx of sodium ions, Na, during depolarization, followed by the efflux of potassium ions, K, during repolarization. This electrical impulse travels along the axon of the presynaptic neuron to the synapse, where it triggers the release of neurotransmitters into the synaptic cleft. These neurotransmitters bind to receptors on the postsynaptic neuron, causing ion channels to open or close, which alters the postsynaptic neuron's membrane potential. If the change is sufficient to reach a threshold, a new action potential is generated in the postsynaptic neuron. This precise electrochemical process enables the transmission of information throughout the brain, with the collective ionic currents generating electromagnetic fields that are vital to neural function. During neural activity, when a single neuron is activated, it generates an excitatory postsynaptic potential, EPSP, at a synapse, leading to local electrical currents within the neuron. These primary currents produce electromagnetic fields around the neuron. Due to the conductive properties of brain tissue, these primary fields can induce secondary currents and fields in the surrounding tissue. These secondary currents and fields can propagate beyond the immediate area, contributing to the brain's overall electrical activity. The resulting electromagnetic signals can be detected by both electroencephalography, EEG, which measures voltage changes at the scalp, and magnetoencephalography, MEG, which detects the magnetic fields generated by these currents. The detection and analysis of these signals allow for insights into brain function, which I will explain further in the next slide of the presentation. In this slide, to understand EEG and MEG foundations, we will connect our discussion to the generated leads and field patterns. As shown in the figure, when a neuron generates an action potential, the resulting electrical activity creates local fields that propagate through brain tissue. The primary current in a population of active neurons gives rise to secondary volume currents, which spread throughout the head. As the skull forms a semi-insulating barrier, the outer currents are considerably weaker. A portion of this current flow between the contact areas of two electrodes, thereby experiencing a resistance and causing the potential difference, according to Ohm's law. In addition, magnetic fields are caused by both the primary and secondary currents. They intersect the cross-sectional area of the pickup coil and contribute to the magnetic flux psi measured by MEG. Understanding the generated fields is essential for interpreting EEG and MEG signals. The patterns of electrical and magnetic fields vary based on the orientation and location of neuronal currents. EEG patterns represent the summed electrical activity on the scalp, while MEG captures magnetic field patterns resulting from neuronal currents flowing perpendicular to the scalp surface. Understanding these field patterns and their interactions between EEG and MEG measurements enhances our ability to interpret the brain's electrical and magnetic activity. Additionally, these concepts bridge the forward and inverse electromagnetic problems, which will be discussed in the next slide. The figure illustrates the structure of the neuro-electromagnetic inverse problem. Neural activity is described physiologically as originating from networks of interacting neurons or neural populations, often modeled as interconnected neural masses. These models predict the current source density, CSD, reflecting activity at a biophysical level. The CSD is then translated into measurements using a model that accounts for the head as a volume conductor and the properties of the sensors. Inverse methods aim to reverse these forward models to reconstruct the CSD or neural sources. Forward problem involves modeling how neural activity generates EEG or MEG signals. By knowing the source configuration, such as the location and orientation of neuronal currents, the forward problem predicts the expected signals at the electrodes or sensors. The central question in neuroelectromagnetics is how to solve the inverse problem. Given measurements of external fields in the brain, how can we trace back to and localize the neuronal sources that generated these fields? This will be discussed in detail in the upcoming video. The inverse problem involves determining the location and orientation of neural sources based on the measured EEG or MEG signals. This problem is more complex because different neural configurations can produce similar signals, requiring advanced algorithms to estimate the most likely sources of the observed data.